So the problem is the same though. What is the least number of flips it takes for Lane to get all the pancakes ordered, largest to smallest, and all burnt side down? So we start with the same starting orientation. If we do the same 2 flips, you'll notice that in the last stack here, our bottom pancake is actually upside down, or as we would call it, burnt side up. So that's unacceptable. So if we go to the next slide, you'll see that it takes 3 more flips. Adam, next slide. It takes 3 more flips to actually get just that one pancake to go from burnt side up to burnt side down, if we start with those original 2 flips. But is this the most efficient way to get the starting permutation to the correct ordering? And as we find out, it's not. We can actually take 3 flips, start by flipping the top pancake only, and then flip the whole stack, and then just the top 3. So Lane's still looking. Okay, so 3 flips instead of 5. So now Trevor's going to talk about something completely unrelated, and we'll make the connection later about how the pancakes fit in with the E. coli.
First, I'd like to say that I've been sick for about a week and a half. So if you hear me squeaking, it's not because I'm going through puberty. All right, so here's recombinase, okay? It's something that Salmonella likes to use to flip around promoters to get different antigens on its flagellum, which is important for them so our immune systems don't freak out and kill them, okay? So there are several things I would like to point out up here. First of which would be these 2 little Hicks sites. One's Hicks L, one's Hicks R. Those are about 26 base pairs in length, and you'll notice that they bracket the DNA segment that's going to be flipped. And in this case, you have an enhancer in there and the promoter. The enhancer that's in there is called the recombinational enhancer, and it's needed for flipping 2. The protein that does most of the work is Hen recombinase. See this little black guy here? It'll bind right here and right here. These are doublets that bind to the Hicks L and Hicks R sites. And you'll notice that this binds in a dimer to the enhancer. You'll see here that the tetramers form when the Hen recombinases form and come together, and then the fisses come in and pull it down like this. You get these loops out here which are stabilized by HU, which is highly prevalent in prokaryotes. And then what happens is you get some strain here, and it makes it susceptible to Hen, which then clips the DNA and then pulls it through. And really all that I want you to take from this, if you're not paying attention to me right now, is the promoter was going this way right here, and if you look down here, it's going that way. Okay? That's about it. All right, so some of the cis-acting elements. You gotta like your cis-acting elements. I told you about Hicks L and Hicks R, which are the things that bracket the DNA that's going to be flipped. And you have recombinational enhancer, which is also part of the DNA. And negative supercooling, which plays a big part, because the negative supercooling basically makes the DNA susceptible, and so it can be cleaved by Hen, which is important. Trans elements, Hen recombinase, the protein that binds and forms the invertisome. FIS, factor IV inversion stimulation. I showed you that. That was the dimer that bind to the enhancer. And then you have heat and stable nucleoid protein, which I showed you in the invertisome, which helps to stabilize that those folds and loops that you saw on the last slide. All right, so burnt pancake implementation. We all know that DNA has directionality to it, right? 5' to 3', things like that. So what kind of pancakes are burnt? Well, promoters are burnt because they can go forward, they can go backwards. Ribosome binding sequences and terminators. So what are some of the things that we're going to need for flipping? Now, as you saw before, we need Hen recombinase, which can be induced by something. And in our case, we use the P-LAC promoter, which a lot of people use. We use IPTG to induce that. Hicks C sites, which I haven't talked about Hicks C sites, but when you have the flipping on the last side between the Hen subunits, you get mixed sites, kind of like you would when you mix restriction sites. And you RE, we weren't really for sure if we were going to need the RE element or not. And how we were going to detect this, which a lot of people, how you detect things in synthetic biology, we use antibiotic resistant genes, or you can use fluorescence. Okay, back to you.
Okay, so hopefully Trevor connected the dots a little bit between the pancake problem and how we were going to solve it. But when we looked back on it, we thought, okay, which one can we solve using the techniques and the research that we just found? And what we found was, if I can get Lane back up here, was that we no longer only had one spatula, but now we have, the waiter can set the stack down and he has 2 spatulas, because you can go into the middle of the stack and flip 2 pancakes from the middle of the stack because of the research that we found. So we didn't just have to flip the top of the stack, we could reach into the middle and flip either a single pancake or a set of 2 pancakes in the middle of the stack. So it changes our problem a little bit. As you can see, now if we got that orientation where just the bottom pancake was flipped, we could take the top 3, lift them up, and flip that bottom pancake over to get the right orientation. Click. Okay, so we started modeling this. We decided to use 3 pancakes because if you look at the sign permutations of the numbers 1, 2, and 3, you get 48 starting possible stacks, so different orientations of how the pancakes can get to lane. And then each stack is 1 flip away from 6 others. So you can either flip the top pancake, the middle pancake, or the bottom pancake individually, or you can take combinations of the first 2 pancakes, flip them, combinations of the second 2, flip them, or you can flip the entire stack. So those are 6 ways you can get from one stack to another stack. What we came up with was the idea to put these 48 stacks on a sphere, starting with the ideal stack, so the 1, 2, 3, the orientation 1, 2, 3, and then with connections to all the stacks that it could reach, with the exception of its complement, is what we were calling it, but the orientation where you would flip the entire stack. And that orientation we put diametrically opposite on a sphere from its complement. So we got down to 24. So on the next slide you'll see it's kind of a representation of the northern hemisphere. If you think of it as a sphere, the X in the middle is the North Pole. The outside line is the equator. And then all the lines represent certain flips, so going from one stack to another stack. Then the colors represent whether you're flipping a single pancake or a set of 2 pancakes. And we're hoping to use this to tell us a little bit about how the E. coli is flipping and why it's flipping maybe the way that it does. So whether it prefers, if you will, to flip a single pancake, or whether it leans more toward flipping a set of 2 pancakes or more. So I think it's back to Trevor.
All right, so our experimental goals, how are we going to pull this off? Well, you take stuff that nature gives you, you isolate it, you change it, and you put it in something else, like E. coli, for instance. So we wanted to design a system in coli that would have a single pancake construct that we could flip. And after we got that to work by seeing either some kind of gene expression through fluorescence or resistance, then we could make our system more complex through 2 pancakes, maybe 3 pancakes, and so on. And then through that, we had the math people testing the pathways and kinetics. Go ahead. All right, so here is a single pancake construct. You'll notice that we have PLAC here, which we can induce with some IPTG. Ribosome binding site, HIN, which if we turn that on, it's going to go over here, and it's going to bind to these 2 Hicks sites. And then it's going to flip this promoter and turn on tetracycline resistance. And it's simple as that. So you'll notice that the starting figure here is going to be sensitive to tetracycline. But if we do promote this, then it will not be no longer, because this is going to flip. All right, so here is a 2 pancake construct. So you'll see that in the starting configuration, you do not have tetracycline resistance. And we have a separate plasmid, which we can induce with HIN. You put HIN in there, we can flip it. And there's different conformations you can have here. One's going to be TET resistant, 4 are going to be RFP expressed RFP. So if this pancake flips right here, it's pretty simple. Then you're going to have RFP expression. If this one flips, then you're going to have TET resistance. And you guys can all see that, so go ahead. All right, so here is a diagram of kind of just reviewing on what I was telling you about how the proteins work in this system. Here's the HIN dimers binding to the Higgs sites. You have the FIS over here binding to the recombinational enhancer. And you'll see that ARIC is repressing PBAD. So if we just throw some arabinose in the system, it's going to take that repressor off, and then you have a promoter which can be flipped if we express HIN in the system. Here is a 4 pancake construct. And if you look at the top slide here, or the top part of the slide, you'll see that you don't have resistance to any of those 3 antibiotics. In the bottom, you'll see that this is flipped, and you can get resistance to all 3 of them. All right, so what are some of the methods that, excuse me, that we use? One of them would just be making synthetic DNA. For instance, we got on the internet, we looked up salmonella in the Hick-C sequence, we ordered oligos with our suffix and prefix on there, and then we annealed the oligos, you cut them, you paste them into the plasmid, you cut them back out, you separate it on polyacrylamide gel, and you get a little pretty gel like this. This is polyacrylamide. This is about 77 base pairs. There's your 100 base pair marker. You know, up here is our plasmid. There's our insert. And it's as simple as that. We use that to make Hick-C. We also made recombinational enhancer and reverse RNA with that method. Next is PCR amplification of natural genes. So how does this work? We all know how PCR works. You take salmonella, you isolate some of the DNA out of them, you find some primers to amplify what you want. So, for instance, in recombinase, you put the prefix on the forward primer, the suffix on the reverse primer, and you make in recombinase. You paste it in, you cut it out. Here's a picture of our tetracycline resistance right here. I believe that is Alvarez gel. And then we made in recombinase that way. We also made the LVA tag. The LVA tag is just a degradation tag, just to control if we want to get rid of in at any part of the process we can with that tag. And we also made 3 antibiotic resistant genes from E. coli with that through that manner. Here's the reversal of parts. So we had to have reverse parts so we could flip them back and forth. We call this the PCR switcheroo. So how we do this, it's exactly the same as the last slide, but instead of putting the prefix on the forward primer, we put the suffix. And on the reverse primer, we put the prefix. Then we amplify the gene, we cut it with XBay and SPE, and we paste it in, cut it back out, run gel. We have a reverse part. It goes in the reverse manner. So through that method, we were able to make reverse PBAD and 3 reverse antibiotic resistant genes. There's a nice little gel there. That's a Alvarez. All right, so what did we help to the registry here? What did we add to it? That's what it's all about, right? Here's 14 basic parts that we made with the help of Davidson. As far as recombination parts, new cloning vectors, antibiotic resistant, genes forward and reverse, and new control elements. We also had intermediates. We had 25 of those we contributed, and 23 different devices. And you'll notice a little code down here. Davidson's red, Missouri Western blue. All right, and here's some of the parts. As far as the insulated plasmid we made, we had recombinase and recombinase with the LVA tag or the degradation tag. We had several antibiotic forward and several antibiotic reverse, PBAD reverse, RFP, things like that.
So in closing, we just wanted to thank, first of all, iGEM for providing us this opportunity and allowing us to come out here and present our research. And then secondly, we want to thank our Davidson colleagues for involving us and for helping us along the way and allowing us to contribute and to just being there for us. And so we appreciate you guys. Thank you.
We'd also like to urge everybody to stick around for Davidson College. Their presentation, like if you're wondering where's the results, a lot of the results from the Davidson presentation and our collaboration on that also is part of the collaboration on ours. Question?
Go ahead.
So does this bacterial strain that you designed, does that solve the problem or does it just represent the problem?
That's what's going to happen next.
It represents it, and then through the flipping, we're hoping that it will solve it for us. If we can sequence it, if that's the right biological term, we can tell what stages it went through to solve it.
Yeah, it's just different versions. Every time we introduce something practical, like Hick-C sites or introduce pancake. So every time there's just different problems we're facing, constructing in any order, any direction, and whatnot. It just poses the problem, and we're hoping to see, which Davidson will talk about, Hick-C, to see if the Henry combinase can solve the problem. Maybe like so.
Any other questions? This is the rest of our team. Eric, Brad, Kelly, Adam, and
We're all
Lynn.
asking if you have one additional question. Did both represent testing process devices? How does that part of collaboration work?
Yes, and actually that's what we were part of testing the concept.
that's what we're hoping. That's how we tried to put it together so that it would extend. I know we just recently had talked about being able to go into a certain part and put the Hicks inside of one of the pancakes so that it would flip not the entire part, but even individual parts of the, say if it was a coding sequence, we could go into the coding sequence, put Hicks inside of there. And so then that would create extra pancakes so we could extend it further. Thank you.
Thank you.